Right, good morning class. If you're in my first period, we kind of went over the first part of the notes, so you guys could fast forward later into the video to where we left off. If you're in my second or third period, excuse me, my third or fourth period, then these notes I don't believe you guys have already done yet. So let's go ahead and get started. So when we're talking about genetics, remember genetics is going to be the study of genes, and that was started in experiments done by a guy named Gregor Mendel. Now, Gregor Mendel had this funny looking hair and funny looking glasses, and maybe I'll pull up a little picture in a little bit. And he worked with pea plants. Now, pea plants, if you guys have ever heard your parents say, oh, you need to eat your peas, those are those little green stuff, right? This is what he used to study heredity. Now, heredity is the passing of traits from parents to their kids, right? Just like you guys inherited some traits from your parents, pea plants are also going to pass on their traits to their offspring. Right. So traits. Traits can be described by their genotype or, and hopefully you guys remember this, by their phenotype. Okay? When we're talking about genotypes, a genotype refers to the allele. Now, allele is just different forms of the same gene. For example, you could have a large head or you could have a small head when we did the bug genetics. You could have red eyes or you could have orange eyes. Same thing from the bugs genetics lab, right? So you could have big A, or you could have big A, little a. A phenotype, on the other hand, you guys need to remember refers to the physical traits. And pH is supposed to help you remember that. So for example, you could have brown eyes, or you could have a disease such as sickle cell anemia. And it looks like we already typed everything here. Let me go ahead and delete that. That way you guys have a chance to write it on your own as we talk about it. Okay. So a genotype can be homozygous or heterozygous. Which one means the same? That you should have said homozygous, right? Examples include big A, or little a, little a. Which one means different? You should have said heterozygous, like big A, little a. And the different alleles, whether it's big or whether it's small, can be classified as dominant or recessive. Do you guys remember which one has the capital letters? Should have said dominance. And hopefully you guys remember the capital letters are going to be dominant, the lowercase letters are going to be recessive. Now Mendel came up with two laws. One law was the law of segregation. And when you guys think of segregation, you probably heard of this in a social studies class or a history class, right? During the civil rights movement, a lot of people fought for their rights to vote, for their rights of speech, right? To be classified as citizens, to be considered equal. And that same idea of segregation, right, we could tie that to separation. Right now we're very, very fortunate to live in a country that has so much diversity. But back in the 1950s, 1940s, way back then, maybe some of your grandparents had to face this segregation, had to face this separation. Okay? But when we're talking about the law of segregation in science, what we're referring to is that Mendel discovered that separate alleles separate into different gametes. And remember, our gametes are just going to be our eggs and sperm. For example, if a man has the genotype big A, little a, he would produce one sperm with big A and one sperm with little a. Okay? Mendel also came up with a second law called the law of independent assortment. Now, the law of independent assortment says that different traits are going to separate into different gametes. For example, the trait of eye color and shoe size are not always inherited together. Okay? So that kind of leads to that genetic diversity that we were kind of talking about. You look at your brothers and sisters and they're not going to be exactly the same as you. Why is that? Well, the reason for that is independent assortment. And if we look at the word assortment, what what four-letter word do you guys see there? You should see the word sort, okay? So they're going to sort differently, independently of each other. Right, so lately we've been using Punnett squares, okay? Remember in my class on Monday or Tuesday, depending on if you're a P day or a G day, okay? We used karyotypes, and karyotypes were looking at genetic disorders like Klinefelter's, remember the guy with the XXY, or Turner syndrome, the girl that only had one X chromosome, or Down syndrome, when we had an extra chromosome number 21. Well, Punnett squares is another thing that you might have seen in 8th grade, 7th grade, or maybe even 6th grade. Punnett squares help solve common genetic problems. So we could write here Punnett squares. In a Punnett square, the gametes from the parental genotype, okay, are combined to look at the genotype of the potential offspring. For example, if you have big A, little a mother that's heterozygous, and a father that's also big A, little a, so he's also heterozygous, they combine, okay? So earlier we said if a dad has big A, little a as his genotype, he will produce one sperm with big A and another sperm with little a. So we see here one sperm with big A, another sperm with little a. Same thing for the mom, one egg with big A and another egg with little a. And then you would fill in the boxes, okay? So for example, here in this box to the left we have a big A, and on top we have another big A, so we'd write capital A, capital A. In this box to the left we have big A, to the right, or to the top we have little a, so we'd write capital A, little a. Same thing down here, and over here, all the way to the left, we have little a, and all the way on top we have little a, so we would have little a, little a. Now what Mendel did was he took different plants and he bred them. And he saw that if you took two parents, and if we look at the genotype, we see that it's a hybrid, right? It's a heterozygote. It has one big A and one little a. So if we said big A codes for tall, and little a codes for short, okay, the big A in the parent would win. So this would be a tall pea plant. The big A also in this parent would win over the little a. The dominant would be the recessive, so it would also be a tall pea plant. So when he combined two tall pea plants, he was surprised when he saw one tall pea plant, two tall pea plants, three tall pea plants, but he also saw one short pea plant, that little a, that homozygous recessive plant. Okay? So this kind of explains why sometimes you see a trait in the parents, and in the kid you see a completely different trait. It's because recessive alleles can be masked by dominant alleles. Right. Now, here is probably the most difficult thing that you guys are going to learn in regards to Punnett squares. We just did something called a monohybrid cross. If you guys remember your vocabulary from biomolecules, a monomer is a single building block. So a monohybrid is looking only at one trait. If you're trying to look at two traits, you would use something called a dihybrid cross. And that, again, that's to look at two traits. Dihybrid crosses use foil, like aluminum foil, F-O-I-L, okay? But that's just going to be an acronym, okay, an abbreviation to represent how we're going to find the 4 different gametes for each parent. So foil, the F in foil, do you guys know what it might be? If the L is last, the F in foil is going to be first. The O in foil is going to be outer. And if the O in foil is outer, the I in foil is going to be inner. So they did this really, really nice thing where they took big B, big T, and little t. Now, if we take the first in the B's, right, it would be big B. And if we took the first in the T's, it would be big T. So we would draw this out, and it would be big B, big T for the first gamete. If we took the outer ones, the one out here and the one out here, we would see that we have big B, and on the outside we have little t. So our second gamete, big B, little t. If we took the inner ones, okay, we'd see that we have little b and we have big T, right, on the inside. So we'd write down little b, big T. And then finally, if we took our last ones, little b and little t, then we have little b, little t. Here's your top 10, and here's a fill in the blank. So what are my expectations? I want you to fill out all 12 blanks, if you haven't done so already, using the word bank or using this. Now these last two blanks, for eukaryotic organisms to mate through sexual reproduction, blank occurs in their gametes. This process is similar to mitosis, and the word is similar to mitosis as well, but it produces cells with how many number of chromosomes? Okay, so these two blanks I don't believe are actually in your word bank, but it's material that we've covered from last week. This blank right here we have not yet covered. Sex cells are also known as somatic cells. And the way I remember that, I think somatic, oh, that starts with an S. Sex, that also starts with an S. Okay? Afterwards, I want you to try these practice problems as well as the packet that I left for the substitute. Now these problems have a step-by-step method. You, on your test, will not have this step-by-step method. So it's important that you guys practice this out. Okay? Here, it tells you, oh, you need to make a key. So if we have big B, what does big B code for? Well, it's dominant, and it's going to code for brown hair. And if we have little b, it's going to be recessive, and it's going to code for blonde hair. And then if we looked at the parents' genotypes, okay, we would say that a mom with blonde hair. So the only way to have blonde hair is if you're homozygous recessive. Is crossed with a heterozygous dad. So hetero, remember, means different. So we're going to have different alleles. We're going to have one big B, which codes for brown, and one little b, which codes for blonde. And remember, we have dominance, so the big B beats out the little b. And then you're going to have to do your Punnett squares. So the mom's gametes, we could put here. One egg's going to have little b, but guess what? The other egg is also going to have little b. And the dad's sperm, one is going to be big B, and one's going to be little b. You're going to do your cross here, okay, just like how we've done. And that's probably the easiest part for you guys. You just look to the left, and you look up top, and you fill it in. But don't forget that there's a question on here. So step 4 says answer the question, and it asks you, what are the possibilities of the baby having blonde hair? So you're going to look at each of these, and you're going to say, oh, this one's going to have brown hair, but maybe the other one's going to have blonde hair. And you're going to write down a percentage or a fraction over here. This right here, your dihybrids, this is going to be super hard. But I know you guys are super smart, so I want you to try it out. If you guys finish with these 4 problems, if you guys finish with your monohybrid practice packet, then you still have these multiple choice problems to do. So you have plenty to do. I will be grading these 4 problems. I will be grading your monohybrid practice, and I will also be grading how far you guys got on the multiple choice. Best of luck today, and I will see you guys next week.